Today's subject is meiosis. Meiosis, the form of cell division by which gametes with half the number of chromosomes are produced. So a diploid has 2N, which are 46 chromosomes. So now they turn into haploids, which are sex cells with 23 chromosomes. Meiosis is sexual reproduction, and this is slightly different from mitosis in that it has two divisions, meiosis 1 and meiosis 2. So this is the fertilization thing, so the fusion of a sperm and egg to form a zygote. A zygote is basically a fertilized egg. So an egg, which is a sex cell, has 23 chromosomes, and a sperm has 23, which forms a zygote, which has 46, which is a cell, basically, your first cell. Meiosis. Sex cells divide to produce gametes, sperm, or eggs. Gametes have half the chromosomes. This makes sense because 23 come from your mom and 23 come from your dad to make one single 46 cell, which then divides with mitosis. Occurs only in gonads, which are the testes or ovaries. In males, this is called spermatogenesis, and in females, it's called oogenesis. I don't know how to pronounce that. Meiosis is similar to mitosis with some chromosomal differences. So we're going to talk about spermatogenesis. So a human sex cell is a diploid cell, 2N equals 46, so 46 chromosomes, which in meiosis 1 turns into 2 with 23. Yeah, and then meiosis 2 turns, like this is spermatogenesis, so you get 4 sperm with 23 chromosomes each. This is meiosis in a mouse. So yeah, this is like on a telescope, so these are some of the steps. That's a parent cell. That's first division. That's second division. And that's the 4 gametes. Interphase 1. Similar to mitosis, interphase. Chromosomes replicate, that's the S phase. Each duplicated chromosome consists of 2 identical sister chromatids attached to their centromeres. Centrioles pairs also replicate. For what they are, for what these things are, you can definitely check out the mitosis video again. Yeah. Interphase 1. More about this. The nucleus and nucleolus are visible under a microscope. So the chromatin is in here. They're bound up in cell membrane, that's the outside, nuclear membrane, that's the inside, and the nucleolus. Meiosis 1. 4 phases. Cell division that reduces the chromosomes number by 1/2. So 4 phases is prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1. Longest and most complex phase consists of 90% of the entire thing. So chromosomes condense. Synapsis occurs. Homologous chromosomes come together to form a tetrad. Tetrad is 2 chromosomes or 4 chromatids, sister and non-sister chromatids. Prophase 1 is synapsis. More about this. So this is 1, and then you get 1. Okay. So these are homologous chromosomes, and these are the sister chromatids bound up. These are tetrads. Homologous chromosomes. Pair of chromosomes, maternal and paternal, that are similar in shape and size. Homologous pairs, tetrads, carry genes controlling the same inherited traits. Each locus position of a gene is in the same position on the homologues. Humans have 23 pairs of homologous chromosomes. Of course, one from one parent and the other. And so, like, these control everything from hair color to, like, how big your eyes are, or, like, if you have earwax, or something like that. But you might be wondering why like you get one set of gene from your mom and one set of gene from your father, but then you would have two sets of traits for each characteristic, like earwax. So this is reflected in the dominant recessive sort of trait. So if there's a dominant trait, it will always trump the recessive trait. The recessive trait only shows up if both your parents have recessive traits on things like earwax and hair color. Have 23 pairs of homologous chromosomes, which are 22 pairs of autosomes, and 1 pair of sex chromosomes. So autosomes control stuff like earwax, whilst 1 pair of sex chromosomes control, karyotype, a method of organizing chromosomes of a cell in relation to number, size, and type. Ooh, look at that. Homologous chromosomes, like for example, that would be the eye color locus. That's a hair color locus. Yeah. That would be the paternal and the maternal. And one would be dominant, one would be recessive, or both are recessive, or both are dominant. Yeah, so humans have 23 sets of homologous chromosomes. Each chromosome is made up of 2 homologs. Yep, we already know this. And of course, what was I gonna say? Okay, let's just move on. In humans, the autosomes are sets 1 to 22. Yeah, and these are the things that you would damage if you get close to radiation for a long period of time. So like the DNA in one of those chromosomes would unravel, and then it would change. Then you get weird children with 4 arms. 21 trisomy, Down syndrome. Can you see the extra 21st chromosome? Is this person male or female? So basically, for Down syndrome, you get one extra 21st chromosome. Yeah, so there should be 2, but now there is 3, which means you get Down syndrome, which makes you I think stupid, or yeah, I don't know. Don't take me on the word for that. So this, in humans, the sex chromosomes are the 23rd set. Sex chromosomes. The sex chromosome code for the sex of the offspring. So if the offspring has 2 X chromosomes, it will be a female. If the offspring has 1 X chromosome and one Y chromosome, it will be a male. So both your mom and your dad randomly give you one of their chromosomes. So my mom would have given me either one of her 2 X chromosomes, and my dad would have given me either an X or a Y. If my dad actually gave me an X, I would be female, and if he gave me the Y, it would be a male. I would be male, but there is no way of deciding. It's just random. These are the X chromosomes, but in males, the Y chromosome is slightly smaller. Boy or a girl, the Y chromosome decides. So that is a big X chromosome, but that shriveled thing is a Y chromosome. Crossing over. Crossing over variation may occur between non-sister chromatids and chiasmata. Crossing over, segments of non-sister chromatids break and reattach to other chromatid. Oop. Chiasma are the sites of crossing over. Good to know. Crossing over variation. The chiasmata, the site of crossing over. These are non-sister chromatids. Yeah. So these are not tetrads. So paternal, maternal, and then they cross. Variation. Another way meiosis makes a lot of different sex cells. Crossing over. Exchange parts of non-sister chromatids, duplicated maternal chromosomes, and duplicated paternal chromosomes so non-sister chromosomes, but these are sister chromatids. So they can tangle up and then they will cross. So these are all given to you by your mother and your father. So, yeah. Crossing over multiplies the already huge number of different gamete types produced by independent assortment. Sex chromosomes. Yeah, we already did this, right? Prophase 1. So we're now getting into like the actual steps of meiosis. So again, spindle fibers form from centrioles, and they will pull your maternal and paternal chromosomes apart. Metaphase 1, shortest phase, tetrads align on the metaphase plate. Independent assortment occurs. Orientation of homologous pairs to poles is random. Variation occurs, and the formula is 2 to the nth power. For example, 2n equals 4. Then n is 2. Thus, the 2 to the square root of 2 for 4 combinations. Metaphase 1. So this is how it happens, or that. So these are the different metaphase plates. Question. In terms of independent assortment, how many different combinations of sperm could a human male produce? Answer. Formula is 2 to the nth power, and human chromosomes, 2n equals 46, so n equals 23. So 2 to the 23rd is about 8 million combinations. Wow. Anaphase 1. Homologous chromosomes separate and move towards the poles. Sister chromatids remain attached at their centromeres. Anaphase 1, so this is what happens. They're being pulled now. Telophase 1, each pole now has haploid set of chromosomes. Cytokinesis occurs and 2 haploid daughter cells are formed. Cytokinesis, basically it splits into 2 cells. Telophase 1, you see there, it starts separating right there. And then we get 2 cells. Meiosis 2 now. No interphase 2, it goes right to the point. Or very short, no more DNA replication. Remember, meiosis 2 is similar to mitosis. Prophase 2, same as prophase in mitosis. This happens again, spindles. Metaphase 2, same as metaphase in mitosis. Anaphase 2, same as anaphase in mitosis. Again, if you want to touch up on what these are, please check out the mitosis video that I also made. Sister chromatids separate. Yeah, very nice. Telophase 2, same as telophase in mitosis. Again, nuclei form, cytokinesis happens. Remember, 4 haploid daughter cells are produced. Gamete is sperm or egg. Telophase 2, this happens. And then we get these. Meiosis, sex cell 2n equals 4, diploid, into n equals 2, and n equals 2 for sperm haploids. Variation, important to population as the raw material for natural selection. This is biology, so I expect that we'll be covering this sooner or later. So the question is, what are the 3 sexual sources of genetic variation? Answer, crossing over, prophase 1, independent assortment, metaphase 1, and random fertilization. Because random fertilization, because there are a million sperms going out for that one egg, so any one of them would have a different variation. Remember, there are 80 million variations, so yeah, it's random. Variation is good. Question. A cell containing 20 chromosomes, a diploid at the beginning of meiosis, would at its completion produce cells containing how many chromosomes? The answer is 10 chromosome haploids. Fertilization, the fusion of a sperm and egg to form a zygote. Zygote is a fertilized egg, 23 egg, and the sperm, the lucky one, will nestle in and you'll get a zygote. Question. A cell containing 40 chromatids at the beginning of meiosis would at its completion produce cells containing how many chromosomes? The answer is 10 chromosomes. nice. And these are the sources. I'll be linking them in the description. So yeah, okay.